The plan for today is to spend a little more time on phylogenetic tree representation and then start the next topic of the history of life on the planet. Reminders, announcements. The first test will be on Monday. There'll be 30 multiple choice questions. There are practice questions available on the course website. This, of course, is in addition to your other opportunities for giving yourself practice questions, which is, of course, a terrific way to study and also to self-test. So you have question assignments on mastering biology. There's also a study area where you can do extra problems should you choose to. And of course, in the textbook, there are a lot of good questions at the end of the chapters. In discussion sessions, you'll be having opportunities to answer questions. And your BioSci peer tutors, any of them here today? I think they were in the earlier step. I want to bring to your attention that the BioSci peer tutors will be having a review session for any of you that want to attend. It's Friday from 5 to 7 in this room. Okay, so that's an opportunity also to help go over the material. What do you need to bring to the test? Just a pencil and a photo ID, that's it. We'll provide Scantrons and the test. A couple of other announcements. This should look familiar to you. This is the top little bit of the overall lecture topic and test schedule for the entire quarter. And I updated it just because I made it more explicit that you do not need to read 29-4 in Bacteria and Archaea, which is going through the key lineages. We'll be pulling out a tiny amount of that information, but you're not responsible for that area as a whole. Anytime I update it, I will let you know in lecture. Also, something else that I put on the course website is a document which is on the web page entitled Grading and Tests. If you go to that particular tab on the course website and scroll way down to the bottom, or close to the bottom, I think you'll hit this item, which is called Items and Textbooks Students Are Not Responsible For. Okay? If you click on it, up opens a PDF document. I'll be adding to this all quarter. So there's various things in here. Chapter 27, you should understand how polyploidy leads to speciation. Don't worry about the terms autopolyploid and allopolyploid. In other words, know the concepts. Don't worry about these two specific terms. So there's various items like this for the different chapters, which I hope you'll find helpful. The individuals in the BioSci lecture podcast captioning initiative asked me to get some feedback back to them, so I'm using this as a clicker question so that I can report back to them. The captioning initiative is that they are providing captions for the lectures, so they want to know if you're using them or not, and if you're not using them, why not? So please pick one of these four. So if you're viewing them or you plan to view them, you should pick A. If you're not viewing them, they want to know why. Pick B if you don't need or want captioning. C, you're not viewing them because you didn't actually know about them. D, you're not viewing them because you don't like something about the captioning specifically. Maybe it's, let's see, question? I'm sorry? Are these the podcasts? So if you go to the website, let's see, let me show you on the, I won't actually take time to go to the website, but if you click on lecture podcasts, if you go to that page, that's where I provide links to the 1 o'clock and 3 o'clock podcasts, which is screen capture and audio capture. In addition, the BioSci captioning initiative takes actually the 3 o'clock one, they don't do this for both. They take the 3 o'clock podcast and then they caption it. And then they put this on a private YouTube channel, and that link and the associated verbiage saying, click here to go to the captioned ones is on that same page, is at the top. So if you didn't know about it, but you wish you knew about it and you plan to use it, then you would pick C, right? Okay? So if you didn't notice that if you go to that page, or maybe you don't look at any of the lecture podcasts anyway, I suppose I should have put that as E. But anyway, or if there's something you don't like about the captioning specifically, maybe the font's too ugly or too small or doesn't correspond to what I'm actually saying, whatever. If there's something about the captioning, pick D. If you just didn't know about it, C. You don't want captioning, B. Or if you're already viewing them, A. If you could all please click in. Everybody clicked in? Fantastic. All right, and I will send this on to the wonderful people that are involved in this captioning initiative. Okay, so about half of you are already viewing them, and then another third aren't viewing them because you actually didn't know about them, but now you do. So now, should you choose to, they are available. If you click on the link on that lecture podcast page, it takes you to a private YouTube channel, and that's where they're posting those. But they're not using the YouTube automatic captioning. They're actually doing real captioning. And as promised, we left off last time talking about sympatric speciation, considering the example of the hawthorn flies and the apple flies, and why this is considered an example of incipient sympatric speciation, meaning that it appears to be a case of speciation caught in action. And I promised a clicker question, and here it is. I'll go ahead and start it up, and then I'll read it to you. All responses count. Do your best to pick the right answer, and then we'll discuss it. So in North America, fly populations that are adapted for laying their eggs in apples rather than hawthorn fruits are diverging into separate species, namely the hawthorn flies and the apple flies, right? Even though apples were introduced just a few hundred years ago. Which of the following facts is most important in making this divergence possible, meaning the divergence between the hawthorn flies and the apple flies? A, these flies are found throughout the range of apples and hawthorns. B, these flies mate once a year. C, these flies mate on the fruit that they lay eggs in. Or D, apple and hawthorn trees are found in the same areas, so the fly populations are sympatric. If you're not sure, chat with your neighbor, think about what is actually facilitating the divergence between these two populations. Chat with your neighbor, click in, and I'll see where you are in 20 seconds or so. Okay, everybody should have figured out, formulated a favorite response, clicked in. Okay. Looks like it's stabilized. Let's see what you guys picked. Here. This is an example, by the way, of a test question on a previous year, from a previous year, so excellent. Half of you got it right, yes. Congratulations. Half of you did not. Let's see, the second favorite answer was actually this one. Now, this may be a true statement that the fly populations are sympatric and they're found in the same areas, but all that means is they're together, which actually does not lead to divergence. In fact, as you may recall, sympatric speciation was considered extremely unlikely and highly controversial for a long period of time because it was hard for scientists to imagine how could you possibly get divergence when they are sympatric. So yes, they are sympatric. This is a true statement. It just isn't an answer to the actual question, which is what's important in divergence. If you have two different populations and they're becoming more different from each other, then what does that mean? Anything where you have less gene flow than between populations then leads to more divergence, more differences accumulating between those two populations. This is a really important item that will lead to speciation, and as half of you recognized, then part of the divergence is hanging out on different fruit types, mating on the same fruit that they then lay eggs on, for example, is hanging out in different areas, different microhabitats. So, nice job. Now, we've discussed phylogenetic trees a little bit already. We're going to continue to build on what you've already learned about and learn some new terminology. Let's go ahead and start right up here. We're going to start by constructing a phylogenetic tree. We're going to just construct a phylogenetic tree for just 4 species. Here they are right here, species 1, 2, 3, 4. And so in order to construct a phylogenetic tree, meaning a hypothesis about the relationship between these 4 species, we need to know something about these species. We need to use some characters. the characters in this particular example, which is from figure 28.1 in your textbook, are molecular characters. And so, in fact, there are 6 characters in this particular example, meaning if we treat these as 6 characters, that means we're treating them each as independent of the others. They're each informative in their own way. As we look at these characters, we're also considering the character states, the different possible values for these characters, and each one has has 4 so-called character states in this particular example. The possibilities are A, C, G, and T for these 4 species. We do see something new in this particular example, which is an outgroup. This is a closely related taxon that's not one of the ones that one is particularly interested in making a phylogenetic tree for. And this is helpful in order to evaluate what the ancestral conditions are. So the character state in the same characters for the outgroup is what is used to infer the ancestral condition. And in practice, one usually are ancestral character states. Ancestral character states for these same characters. In practice, one typically uses more than one outgroup for further corroboration. In this very simple example, we're just going to use one outgroup and assume that this is completely informative about the ancestral condition. So let's go ahead then and make a phylogenetic tree. So I took that same information, just sort of made it a bit smaller so that we would have room to write here. So let's now figure out what the relationship could be between these 4 species, and we'll put the outgroup in there as well. So we have these 6 different characters. We can focus on nucleotide number 3 and nucleotide number 5, because these are the ones where we actually see some differences then among the 4 different species. If we look at the other ones, they're actually the same. So for example, nucleotide number 1 is the same in all of them. But if we focus on 3 and 5, we start to see some differences. And specifically, species 1 and 2 have C at nucleotide location number 3, which is different from the inferred ancestral condition. So this is something that's shared by 1 and 2. And not by the others. So this sounds like species 1 and 2 are sister groups. So let's go ahead and make them sister groups. Similarly, 3 and 4 share G in nucleotide position 5, which is different from the inferred ancestral condition. And so let's go ahead and make species 3 and 4 species group, sister taxa as well in this phylogenetic tree. And then we can go ahead and add the outgroup here. And here we have one possible phylogenetic tree for showing the possible relationship between these 4 species. Now, if we then use this phylogenetic tree to superimpose changes that may have occurred in evolutionary time, let's look at our data matrix here to figure out what happened when. Again, we're going to assume that we can use our outgroup as an unambiguous identifier of the ancestral condition, right? So ancestral character states, I'll write that up here again just to remind you. And so because we're focusing on 3 and 5, I'm not going to write out all 6 again. I'll just put 3 to 5 here is A and C, right? Meaning that at the common ancestor of the outgroup and the 4 species of interest, we're assuming that the most recent common ancestor of all 5 of those taxa together at this point here had A at nucleotide position 3 and C at nucleotide position 5. Right, does that make sense to everybody? Now, if we consider what is going on here with species 1 and 2, we know that at number 3 we have C, and at number 5 we have C. So if we write those down, then what we have for positions 3 and 5, then we notice that compared to the ancestral condition, we've had a single change in nucleotide 3. Because that's a shared condition, a logical place to put this in this phylogenetic tree would be right here at position number 3. If we have a substitution or a change from A to C at nucleotide position number 3, then we end up with the C at both nucleotide 3 and 5 for both species 1 and 2. Is everybody following that? Right? In the absence of a change, you just assume that it stays the same. Now, these vertical tick marks, such as the one that I just drew right here, a vertical tick mark is a handy way of identifying on a phylogenetic tree where a change took place. That means at this specific branch, somewhere between this point, this divergence point and this divergence point right here. Okay, we've also had another change, right? In species 3 and 4, we have AG at these same two locations, right? And if we compare this to the ancestral condition, looks like they've retained A at nucleotide position number 3, where we see a change is nucleotide position 5. So again, similarly, we can hypothesize that at nucleotide position number 5, we had a transition from C to G. So if we then make this assumption, then we end up with, for this specific phylogenetic tree, we end up with these relationships and these evolutionary changes through time. Okay? And you might be thinking, well, this isn't the only tree that's possible, right? And that's quite true. Let's consider another possible example, which is why I gave you two sheets that are exactly identical like this in your note-taking helper, so you could draw a different one on this other page if you so choose. Let's do another one, again, reminding ourselves that these are the locations where the nucleotides are different. Let's draw another tree, okay? So now we're going to make species 4 and 2 sister taxa instead of 3 and 4, and we're going to go ahead and put, let's see, 3 here and 1 here, and then we'll add in our out group. And there we go. Different tree. Again, reminding ourselves of the character states. We're still assuming that at this point we have the ancestral condition for 3 and 5 will be A and C. Let me go ahead and change color. I think that'll make it easier to make it stand out. And if we consider the conditions for 3 and 5, reminding ourselves of what they are. 4, we've got A and G. Number 2 here, we've got C and C. We've got A and G. And we've got C and C, right? So, how can we explain these differences? If we start with species 1, then, this is a lineage that is descending from something that had A at nucleotide 3 and C at nucleotide 5, and now we end up with C at both, which means what? Some point along the evolutionary history of lineage that led to species 1, we had at position number 3, we had an A turning into a C, right? And that would be completely consistent with this. How about species number 3? Again, following the logic here, we have A, C here. Looks like we have a change in nucleotide number 5 somewhere along here. So here we go, number 5, we have a change from C to G. Number 2, looks like a change at number 3 from A to C. And then here at this point, again, a nucleotide position number 5, we've got a change from C to G. Does the logic make sense? You understand how you could start with a phylogenetic tree like this and say, if in fact we know that they have these character states, if we start at this point, how can we get to these endpoints from that starting point given this phylogenetic hypothesis, this hypothesis about the relatedness between these species and this hypothesis for the evolutionary history of the group. Okay, so we have two hypotheses, right? We have this one, we have the earlier one. Well, how do we know which one is right? How do we know which one is better? What criteria do we use for selecting one or the other? When one is selecting a preferred tree, one uses parsimony, meaning the simplest possible explanation, the fewest number of changes or steps that is necessary. This is actually a more general philosophical approach to many different kinds of problems. It also has the name in philosophy of Occam's razor, the general statement that a less convoluted, simpler explanation is usually preferred over a more complex one. And so I think an example of this which might make more sense to you, is if you consider the relationship between two variables x and y, and you go out and you take data because you're interested in the relationship between x and y, and your data look like this. You end up with this relationship between x and y. Then you can infer, you can make a prediction about the relationship between x and y. So for example, if I put the blue line that passes through all of these points, that is one possible explanation for the relationship between x and y. I'll get a different color. It is, of course, possible that the relationship is something different, that the relationship is actually like this. See? Notice that this particular trend actually passes through every single point just as well, right? There we are. It went through every single point. Both the green explanation and the blue explanation went through every single point. Either one could be true. Either one fits the data exactly as well as the other. But the reason that the blue explanation would usually, under almost all circumstances, be preferred over the green one is that it's simpler. It requires a lot less unnecessary complexity. And this is the same logic or philosophy that goes behind selection of the preferred phylogenetic tree is one that assumes the smallest number of changes. This doesn't necessarily mean that it's correct, but it's the simplest explanation for the data that went into construction of that phylogenetic tree. One can always further test the tree by collecting more data and seeing if it further corroborates the tree or not. So we then, armed with the principle of parsimony, if we go back then and look at our trees, let's evaluate these in terms of how many changes are necessary. If we look at this one, for example, for this particular tree and these particular changes, we have to invoke 1, 2, 3, 4 evolutionary changes in order to make this scenario match the actual data. If we look at our earlier first phylogenetic tree, if we do count the same thing, the number of changes, we see that there are 2 changes. And therefore, of these 2 particular trees, then the first one here is more parsimonious, and it would be preferred as a simpler, more direct, more compelling explanation for the evolutionary history of this group. So that's how one uses parsimony. Now, this actually leads us then to a few more terms which are explained in figure 28.2, which is in front of you right now. So here's one of the terms, and this is the definition of homology. And homology is defined as similarity in organisms that is due to or caused by common ancestry. So in the example phylogenetic tree that is in front of you right now, we have something happen, some change again. Look at the vertical tick mark that indicates something changed. And this is now shared by all further descendants in that lineage. You can see we're uniformly orange later in time in that lineage that continued to branch and diverge a bit into time. So all of the downstream individuals share A. So these 4 terminal taxa here all share characteristic A, whatever that is. But this shared characteristic A is because of common ancestry. They all received A from their ancestors. Now, if we go back to those earlier phylogenetic trees we were just looking at, we can see why choice of different tree implies different things about whether or not something is homologous or not. Let's go back to this one. Notice this particular trait right here. The trait that species 1 and species 2 both have a C right here at position 3. This would be an example of a homologous trait in species 1 and 2 because it is a result of shared ancestry. See, there's the common ancestor right here. We had a change in nucleotide position, A turning into C at that point. So this would be homologous. If we look at our alternative explanation here, again, here's C and C at position 3, but now it's no longer homologous. So these are similar, but not homologous because the similarity is not caused by homologous. Because this similarity having C at nucleotide position 3 is not caused by shared ancestry. In fact, This is a result of two completely independent evolutionary events, each of which independently generated C at position 3. So the phylogenetic tree is incredibly important and as a determinant for evaluating whether or not something is homologous or not. So back here, in this particular case then, this is clearly homologous. Now here's a really important term for you, a monophyletic group. This is a very important concept in phylogenetic trees. It is defined as a unit that includes an ancestral population and all of the descendants. And so how one can actually figure out if one has a monophyletic group when one is looking at a tree like this is that it passes the so-called one SNP test. What does that mean? Well, if you're a gardener, let's see, one SNP test. Look at your phylogenetic tree. You've got this branching, bushy structure here. They could be quite bushy. If one comes in with a pair of clippers and prunes the bush and makes just one snip somewhere, whatever falls off is a monophyletic group. If you can generate a group with just one snip, that's a monophyletic group. So in this particular one that is shown here in orange, there's where we snip right here. This bit falls off. That's a monophyletic group. Okay? We could also snip right here. This is also a monophyletic group. It is a different monophyletic group. Snip right here. This is also a monophyletic group. Any group that includes a common ancestral lineage and all the descendants is a monophyletic group. Now, the last term that we need to wrap our minds around before we leave this particular slide is this term right here, synapomorphy. And that just simply refers to a trait that is shared by a particular monophyletic group. So the example here is A, which is a trait shared by all members of this monophyletic group. It is derived, just simply meaning that it is not shared by the outgroup or the other distant relatives shown in light gray in this particular diagram. An example will probably make that clear in just a minute. So, and of course, not all similarities are caused by shared ancestry. In fact, when we just walked through that example, we had the two different phylogenetic hypotheses, one of which resulted in an interpretation of homology for nucleotide 3 in species 1 and 2 being homologous, and the other phylogenetic tree that it was not homologous. If it's not homologous, then this is an example of homoplasy. So this is kind of like the opposite of homologous. So not homologous, or homoplasy, not homology. So this is similarity due to reasons other than shared common ancestry. So the reasons can be quite different, but in this particular case, and this is like the second phylogenetic tree, for example, we were looking at earlier, in this particular case we have B arising 2 times independently, and therefore the fact that this particular taxon at the tip here and this particular taxon at the tip there, they share B, but they don't share B because they both have inherited B from a common ancestor, but there were 2 completely different evolutionary origins of B. You can also consider this an example of convergent evolution, where you end up with the same thing in different lineages independently. And a group like this, which is not monophyletic, so just this taxon here and this particular taxon here, this is not a monophyletic group. In fact, you can see immediately it flunks the no SNP test, not monophyletic group, because to generate a group that just includes the purple taxa without anything else, we'd have to do 2 SNPs, right? We'd have to do 1 SNP here and 1 SNP there to generate that group. So you can talk about a set that includes those 2 taxa, but it's not a monophyletic group. Monophyletic groups are generally preferred because they're a clearer, distinct evolutionary lineage. Here's an example of homoplasy. This is figure 28.3 from your textbook, and shows how remarkably similar dolphins, which is a kind of marine mammal shown in the top here, how similar they are to an extinct group called the ichthyosaurs. The ichthyosaurs and the dolphins both are streamlined. They have fins and flippers. They have long beaks or noses with, or long jaws with sharp teeth and so on. And they're very streamlined. They're fast swimmers. They're very similar in shape. But these characteristics, this list of characteristics, streamlining and long jaws and sharp teeth and etc., are considered examples of convergent evolution, because if one looks at a phylogenetic tree that shows the presumed relationships between dolphins and ichthyosaurs, you can see that dolphins as a mammal are grouped here within all the other mammals because of all the other characteristics they share with all the other mammals, and ichthyosaurs are much more closely related to lizards and other vertebrates, non-mammalian vertebrates. And so these would then be considered, these character states would be considered an example of homoplasy or independent evolution of these particular traits in these particular groups. Now note, of course, that if the only traits that we used to generate this tree in the first place were streamlined shape and fins or flippers and long jaws and sharp teeth and so on, they would end up as sister taxa, right? But if one uses a very large number of other kinds of traits, then this allows one to pull out what are truly homologous traits and what are truly examples of homoplasy, as shown here. And then finally, the last panel of figure 28.2 just shows another different kind of non-monophyletic group, non-monophyletic. The so-called paraphyletic. So this is slightly different than the polyphyletic. This is just simply a group that at first glance looks monophyletic if you look at this blue bit, but then if you look very closely, then you can see it does not in fact include this group right here. And so if one was going to pull out all the taxa represented in blue, meaning not this one right here, then one would have to do two SNPs, right? One SNP there, one SNP there. So this, including all of them, is an example of a monophyletic group. So you'll see this throughout the quarter as we talk about different groups and which ones are monophyletic and which ones are not, and why are fish, for example, considered paraphyletic. So it's not considered a natural grouping. So a nice example of this is actually seen in our current understanding of the relationship of whales to the other mammalian taxa. This is figure 28.4 in front of you right now, and shows the relationship, the old phylogeny, showing how whales related to other mammals. So we've got whales up here at the top, and then we have a whole bunch of animals here, camels and pigs and hippos and deer and cows and so on, all of which share this particular characteristic, the so-called astragalus, which is the name of this particular bone seen in the ankle and is seen in camels and pigs and etc. It's not seen in whales or horses or rhinos or other mammals. So this would be an example of what? A synapomorphy that identifies this as a separate monophyletic group that does not include the whales. As scientists studied whales more, they noticed that whales really seem to be quite similar to hippos in many different ways. There's hippos way down there. In this phylogenetic tree, they are not particularly closely related. But one of the characters that was very useful in pulling out the relationships between whales and other mammals was various molecular characters, including these transposable elements with the acronym SINE, S-I-N-E. The data are shown in figure 28.4 in your textbook, which are shown right here. Over here on the left-hand side, we have a bunch of different mammals, so cows and deers and whales and hippos and so on. And these numbers across the top here just simply show locations for these transposable elements. And the Scientific Illustrator has indicated the very strong similarities between whales and hippos that are seen in this particular characteristic. Now, this is not the only basis for seeing, identifying whales and hippos as fairly closely related. This is just one example of many. And so this is then why the new phylogenetic, or the more recent phylogenetic hypothesis of how whales fit into the other mammals is shown here on the right. Here's the old one that we were looking at before, and you can see that whales now are in a new position. Whales are now shown as a sister group to the hippos, even though they have the loss of this particular bone. So this is an example where if one included only the mammalian groups that included this particular ankle bone, one would end up with a non-phylogenetic group, right? Not all of the descendants. And in fact, whales are properly included in this group, even though they don't share the synapomorphy because they have lost this particular character. So the gain and loss of particular characters then can make it very difficult for inferring phylogenetic trees, which is why scientists tend to use very large numbers of characters in the first place, so that one can be as sure as possible about how robust your particular tree is. So if you understood that then, you should be able to wrap your minds around this particular question. So consider you have 4 species W, X, Y, and Z, and you're interested in generating a phylogeny of these species. Here's a handy table for your convenience showing the species as columns, and we have 3 traits for these 4 species here, and these show the character states for each of the species. So for example, trait 1, here are the character states for trait 1 for the 4 different species, trait 2 for the 4 different species, and trait 3 for the 4 different species. And I want you to consider which of the following traits is most useful in generating a phylogeny for these 4 species. Do you think trait 1 is most useful? Do you think trait 2 is most useful? Do you think trait 3 is most useful? Or do you think they're all equally useful? Why don't you consider this question and chat with your neighbor for 20 seconds or so, and then I'll check back in with you. Okay, everybody should have figured this out. Clicked in your favorite answer. Let's see what you guys think. This is another example, by the way, of a former test question that was on a previous exam in this class in an earlier year. Okay, looks like everybody has answered this one. Let's check this out. All right, what is the best answer? Interesting. About a third of you got this right. Good for you. Okay, so what led the rest of you astray? I mean, it's hard to believe that more information couldn't be more useful, right? But if we specifically think about understanding the relationship just between these 4 species, let's check out trait 1 first here. Notice that trait 1, it's exactly the same character state in every single one. There is no signal here. There is no information about the actual relatedness between W and X or Y versus Z or anything. Okay, let's look at trait 3 next. How about this one? That's C, D, E, F. It's different for every single species. Again, no signal in terms of understanding the relationship between these 4, unless one also is going to assume some sort of relationship between these different character states. But in the absence of that, they're just completely different from each other. There's no signal. There's no information. Certainly they might be informative in understanding how these 4 species perhaps relate to other taxa. But just resolving the relationship between those 4, neither trait 1 nor trait 3 is useful at all. Trait 2 is not completely useful, but at least it gives us a little bit of a signal here. It suggests that W and X are more closely related to each other, and Y and Z, right? Following this tiny snippet of information, then this is at least how that is going. But it makes a more important, more general point, I think, is that for character states that are exactly the same, or conversely, completely different for every single taxon, it's not helpful then in pulling out the relationships. It's the shared characters that are the useful pieces of information. Okay, let's try another one. This is much more simple, and I bet you'll all get this one right. Let's try this one then. consider some insects, specifically some beetles and flies that have antler-like structures on their heads, much like male deer do. The existence of antlers in beetle, flies, so these two insects, and also deer, this mammal species with strong male-male competition would be an example of what? And in fact, the presence of antlers is associated with male-male competition in these different groups. So what is this an example of? Homoplazy, synapomorphy, homology, parsimony? What do you think? Chat with your neighbor, click in, and I'll see what you guys think. Another example of a former test question, by the way. Okay, presumably everybody figured this out? Clicked in? Last one? Okay, everybody's in. Let's see. Awesome, look at that. I knew you could do it. Excellent. Yeah, this is a perfect example of homoplazy. These insects are not particularly closely related to this mammal group, the deer. So the existence of similar structures is absolutely an example of convergent evolution. So if you got that wrong, make sure you review the meaning of these terms. Now we're going to switch gears a little tiny bit. Yes, thank you. And consider the geological, biological, sorry for the weird word wrap thing going on here, geological, biological history of the Earth. This is figure 28.6 in front of you right now and shows a timeline in the way that geological times are often shown, which is the present time or now, or at least more recent up at the top, and earlier times further down, sort of evocative of what happens when you're digging down and you get older and older and older sediments as you go deeper down. Anyway, so time is vertical in timelines like this. And you can note a couple of things here. If you can read this, this is about 4.6 billion years before present. Billion years before present at this point right here. So the Earth is really old. You should certainly know this number. Do I care if you know 4.6 versus 4.5? No, of course not. But you should know it's 4.6 billion, not 4.6 thousand, 4.6, Google, whatever. 4.6 billion years. And it's such a vast amount of time that it's subdivided in a nested hierarchical subset kind of way, just as taxonomic classification systems are hierarchical and nested. But when we talk about time, geological time, the largest category, most inclusive categories are called eons. And so we have 4 eons. Here they are, 1, 2, 3, and then 4. The fourth one that we're in right now is written sideways here. Eons are divided into eras are divided into periods, and so on. There's a ton of names. I know when you hit this part, you probably thought, I am completely overwhelmed by all the names of all of these different eons and eras and periods and epochs and so on. The first thing I want to reassure you is you are going to be responsible for a minuscule number of these. In fact, that's in the document that I showed you at the beginning of this lecture. And that I'll make sure to make that perfectly clear. So we're just going to learn a couple of specific points to use then as comparisons. But let's start by considering down here 4.6 billion years before present. We're talking about the formation of the solar system, the first appearance of our planet. At this point, it was extremely hot. We have origin of life occurring about 1 billion years later, and so first there had to be a cooling period. So there's cooling going on down here earlier, and earlier on we considered the cell theory and how cells come from cells and came from earlier cells, and also that species come from other species, and so on. Well, now we finally hit the point in time where we no longer evoke those earlier species and those earlier cells, because we're starting now, we're considering the very origin of life. This was such a long time ago that, of course, there is not a lot of direct evidence. And so much of the hypothesizing then is based on the physical and chemical laws, as well as the information that we have about the conditions on the early planet. We're not going to spend very much time on this, but we'll consider just one really important part of this, which is going from a completely lifeless planet to cellular life. The first step, presumably, is at least ending up with formation of organic compounds. This is sometimes referred to as considering how chemical evolution could have occurred. How could complex organic compounds appear on the planet without organisms there to actually synthesize these organic compounds in the first place? Now, in the 1920s and 1930s, a number of different scientists, including Haldane and Oparin, who were experts in planetary chemistry, considered that the conditions on the very, very early Earth, which are completely different from the conditions now, could in fact result possibly in the spontaneous formation of organic compounds from very simple precursors. So why was the early Earth so different? Why were the atmospheric conditions so completely different? Well, because after the Earth cooled down a lot, there was a lot of volcanic outgassing, and so compounds like methane, ammonia, hydrogen, sulfur dioxide were very common in the atmosphere, although now they're not. When we think of the major components of the current day atmosphere, what do you think of? You think of nitrogen and oxygen and so on. Completely different molecules in different proportions than made up the very, very early Earth. Now, what happened in the 1950s is two scientists called Miller and Urey ended up addressing this interesting idea directly experimentally. Now, at this point in time, Urey had actually already won a Nobel Prize in chemistry. 20 years earlier, he had won his Nobel Prize for his work in isolating isotopes of different chemical compounds from each other. He was an expert in planetary chemistry, and he was interested in testing these ideas about formation of organic compounds in the laboratory. So he did this with his student, Miller. This is how they did it. This is what the apparatus looked like. It was a very simple setup and completely enclosed in glass. And what they did was they set up conditions that were similar to what were hypothesized for existing on the early Earth. So we do have water. Water's not shown here. Obviously, you know what water looks like, H2O. Ammonia, right? There's ammonia right here. Methane, another compound that was presumably very common in the early atmosphere, and also hydrogen, H2. If you start with this gas mixture, enclose it in glass tubing like this, and then heat a glass flask like this, causing then what? The water to vaporize, moving around this system here. And then over here in the upper right, you can see a couple of wires. Here they would have simulated lightning, where they would put a voltage drop across this, and zip, a spark would go from one to another, zapping the atmospheric compounds then in the gas mixture, and this would go round and round and round and round. So they would supply occasional heating of the water, sparks, and then after a week, they opened it up and they looked to see what was in there. And what did they find? They found amino acids, they found sugars, they found a variety of different compounds. This has been now repeated in a number of different times with different ratios, different conditions, different lightning bolt frequencies and so on. And you can produce a number of different organic compounds in a mixture like this, which is terrific evidence then for how one could start with organic compounds then in the absence of life forms with the right conditions. So I'll go ahead and stop there, and on Friday then we'll take up where I'm leaving off, and perhaps I'll see some of you at office hours tomorrow.